Hello, and welcome to our virtual field trip. I'm Teresa, one of the board members at Washington's National Park Fund, and I am proud to be your host today. Before we start, there are a few housekeeping items. This trip is 45 minutes with a presentation from our speakers and a Q&A at the end. Please feel free to enter into any questions in the box as we go. It has closed captioning enabled, which you can access at the bottom. We welcome any feedback as we work to make these trips as accessible as possible. For those new to Washington National Park Fund, also known as WNPF, we are the official nonprofit partner for Mount Rainier, North Cascades, and Olympic National Parks. We have the honor to work closely with these parks and their park leadership as we work to fund 30 to 40 priority projects each year. And just this past year, we awarded over $1.5 million to projects that fall into 4 core areas: advancing science and research, improving visitor experience, expanding volunteerism and stewardship, and embracing inclusion. Today, we are so excited to highlight a project that blends together research and volunteerism at Mount Rainier National Park. The Science for All project focuses on unraveling the mysteries of the rare carnivores that call the Cascade Range home. I'm looking forward to sharing this project and how supporters have made an impact for the park. Let's dive in, shall we? We have 2 amazing speakers with us today to cover this incredible project. First, Sallie Beavers is the Chief of Natural and Cultural Resources at Mount Rainier National Park. Sallie is an ecologist with nearly 3 decades of federal service and 24 years with the National Park Service. Prior to joining the team at Mount Rainier in 2022, she served the national parks on the west side of the Big Island of Hawaii. And Jocelyn Akins is the founder and executive director of the Cascade Carnivore project. The shoestring effort started back in 2008, focuses on rare carnivores in the Cascades, and has now grown to a board of directors, staff, field crew, and over 70 volunteers. Jocelyn loves employing genetic and ecological tools to better understand how carnivores and their habitat are faring in the face of climate change. We're so excited to dive in to learn more about this amazing project. So, friends, take it away.
Oops. Oops,
Sallie,
sorry.
if you'd like to
Trying to find my various buttons. Apologies, everyone. Hi, everyone. I'm Sallie Beavers, and I am the division lead for Natural and Cultural Resources here at Mount Rainier. And I'm really excited and pleased to be spending time with all of you today to share with you the exciting work that we're doing in partnership with Cascades Carnivore Project, with support from Washington's National Park Fund. So Mount Rainier's Science for All project is an innovative collaboration between the park's Natural and Cultural Resources program and the Cascades Carnivore project. It's a community-driven science initiative, and it's focused on understanding the behavior, habitat, and conservation needs of the Cascade red fox. And through our partnership with Jocelyn and the Cascades Carnivore project, we're delving deeper into understanding the habits and habitats of the fox and how climate change and human activities affect the species inside the park. We collaborate closely with Jocelyn and CCP because of their invaluable expertise and their long-term research on imperiled carnivores throughout the Cascades region. And that last point is key. Carnivore research within the park is critical to park management, but it's really the regional context that's vital to informing our park conservation efforts and understanding the big picture for parks' rare carnivores. And I want you to know how grateful we are to be talking to you, because Washington National Park Fund plays a pivotal role in supporting this and other work throughout the park. And I can't overstate the positive impact that donor contributions have in providing essential funding that facilitates studies to inform management of alpine wildlife in the park and statewide. And nor can I overstate how volunteer community science amplifies the data collection beyond what we can do ourselves. And in fact, community scientists participating in collecting the data that supported Washington Department of Fish and Wildlife's determination to list the Cascade red fox was really key in 2023. So over the years, hundreds of thousands of dollars of WNPF donor funds have supported park science needs. And those funds go to survey supplies, data sampling gear, data analysis, remote camera stations, vehicle costs, wages for seasonal technicians, and beyond. And we're especially grateful for the support that this project has received from WNPF and all our partners. And I just celebrated my 24th anniversary with the National Park Service last month. And one of the most rewarding parts of my job is sharing the coming together of communities to safeguard our parks and ensure that they remain extraordinary and resilient. So with that, I'll pass it on to Jocelyn, who's going to talk about the work happening in the field and how volunteers can support her work.
Thanks so much, Sallie. All right, let's get started. Awesome. Thanks everyone for being here. I'm excited to share what we've been up to for a long time working in partnership with the National Park Service. So today I'm going to largely be talking about 2 carnivores, and there'll be a little sneak peek at the end on a third that we also study. But today I'll be talking about the Cascade red fox and the wolverine, both of which occur in Mount Rainier National Park in high elevation habitats. So our work in partnership with the park, it all began as a small project in 2008 to study the return of wolverines to the South Cascades generally, so south of I-90. And it started on Mount Adams to the south. And as Teresa mentioned, 15 years later, here we are with a small but mighty nonprofit and an amazing team with a board of directors, a small staff, an incredible field crew, and now we're actually up to over 80 volunteers. And our mission is to study and protect rare carnivores. And what I mean by that is that we conduct rigorous research projects to understand the status, how the carnivore populations are doing, and then also to understand and learn what is threatening them if they are in decline, which we often find with these rare species. And then the protect part, that is not something that we can do alone. We do it in partnership. We have great allies and partners, including the National Park Service and all these other groups. So it's very much a large team effort to work to change policy, to inform management plans, to identify priorities that may recover the species of interest and restore their habitats. So I just want to get started by telling you a little bit about myself. I am a conservation geneticist. I have a PhD from the University of California at Davis. And conservation genetics, that's just a fancy way of saying that I like to collect the things that animals leave behind. And what I'm talking about is DNA largely, so scat and hair, even urine samples that you can find in the snow. And I really love studying animals that are hard to find and live in these remote wild places, species that you don't normally see. And it's hard to understand what is going on with them, how their populations are doing. I've worked in a few other national parks, including Yellowstone National Park and Grand Teton National Park, studying grizzly bears, and I also studied wolverines there, and that is really where my love for the wolverine came from. And I think this love and interest in studying carnivores and working on conservation efforts to protect them is from a lifetime of hiking in the Cascade Range. This is me and my dad on Mount Baker to the north. I've been hiking in these Cascade Mountains my whole life, and I just feel very fortunate to be able to spend my career in the mountains as well. But why care about carnivore conservation? We know that carnivores play a critical role in the ecosystem, keeping prey populations in check, and this in turn keeps the vegetation and the landscape healthy. And through climate change, habitat fragmentation, and increased recreation, as humans, we continue to leave our mark on the landscape. And when I came to the Cascades, I noticed that there was very little understanding of some of the rarest species, the ones that are so hard to find. And for me, having wild creatures on the landscape in our mountains it just gives you a sense that the ecosystems are intact and healthy, and that it's a wild, wondrous place to be. And so our research spends time getting out there and learning about how these populations are doing. We have been studying the return of the wolverine to the Cascades since 2008. Wolverines were wiped out from Washington and Oregon by about the 1920s. And this was due to predator control programs that were aimed at wolves, at the larger carnivores, wolves and bears, and coyotes, and also to overharvest. And there was a lot of incidental effects of these predator control programs. And the wolverine was largely declined due to the poisoning programs that happened up until just recent decades. But what's so wonderful about the wolverine is that it returned naturally on its own, and it came from British Columbia and Alberta, returning into Washington. In about the 1990s, wolverine researchers noticed that they had returned to the landscape. But there were still no wolverines on Mount Rainier. So this slide is from the verifiable database of wolverine records that's maintained by the U.S. Forest Service. And you can see here, this is a time period of 5 years from 1996 to 2000. And there's only 3 verifiable observations. So this was just the start of data collection showing that they had returned to the Cascades. In 2001 to 5 period, you notice that there's an individual down south here. This is Mount Adams. So it had skipped Mount Rainier, which is right here, but you could see that there was a few more individuals and they were slowly expanding their distribution. Now, skipping forward to 2016 to 2019, you can see that there really has been a lot more observations of wolverines in the Cascades, including, these are from our research, they're showing up at Mount Rainier for the first time. So you can detect wolverines from camera stations, but it's difficult to identify individuals, especially to see if there's females that have returned to a region. It's hard to know how wolverines are doing when they roam these very large areas and you can't distinguish individuals or document reproduction. So you don't know if you have like a lone dispersing male, or if you have a new population establishing itself. These are some of our early wolverine photographs. They're all from south of Mount Rainier. And you can see that there's, I should say, every wolverine has a unique chest mark. It's called its chest blaze, and it's a white pattern. It's a white blaze on its throat and chest, and it's unique to each individual. And so you can identify wolverines that way. And in these photos, it's hard to see, they're not high quality, but in some of them you could tell that you have the same individual, the little white patch under its throat, but others it was really unclear, and we didn't know from these sets of photos that we were accumulating whether we had one or more individuals. So in 2016, we shifted gears and we deployed a large spatial array of monitoring stations across the whole South Cascades region, from Interstate 90 to the Columbia River, including about 6 stations in Mount Rainier National Park. This is a diagram that shows what these crazy stations look like. So they're a little elaborate. They take time to set up in the field. But the cool thing is that once you have them set, then you can learn a lot about the wolverine. And primarily you can learn the individual identity of the wolverine by seeing that unique chest pattern on photographs. The stations also collect hair samples, so you can send those for DNA analysis, and you can also confirm the individual ID. You can learn its sex, and you can also learn the population of origin. So where did it come from? Is it part of a Cascades population? Is it a migrant that's come from elsewhere? And so the way these stations work is you have 2 trees, we climb up them, and we sling this cable across them, and we hang a piece of bait down onto this structure that we build. And the structure is called the run pole. And I like to liken it to on a pirate ship when you're forced to walk the plank. So the wolverine, when it tries to get the bait, it might come up the tree. No, it's too far. But if it walks out on the plank and climbs up these uprights, which are made of 2x2 wood, then it can access the bait. And as it's looking up and climbing up, the camera on the other tree is taking photographs. And it's also walking through these lower uprights, which have wire brushes on them that collect hair samples. We usually have a second camera you can see down low or on another tree to get a different angle. So this is the view in the field. It's hard to see, but here is the bait hanging here. So this is an elk hindquarter, and this is the plank that the wolverine walks out on. You can see we climb up the trees to set the cable, and the reason that we set them high is that you can leave them to overwinter. So in some of the more remote parts of the Cascade where it's hard or impossible to get to, it certainly in a day, we'll set stations that remain out and can be active all winter. The batteries in the camera are lithium and they last all winter. And we've shifted to a large bone so that this bone stays, it's the wolverine is unable to break it, it's unable to take it away, and it lasts all winter. Here are the hair brushes. So, and we also experimented, and wildlife research, especially wolverine research, is very much in experimentation. It's half art and invention, I feel like at times, but these alligator clips. One alligator clip is holding another one open, and as the wolverine walks by them, it pulls a hair sample, and then you can send that one to the lab, and then another time, if there's another wolverine, you get a separate sample. So can you see here that these two wolverines are different individuals? So on the left, we have less white. It's almost a boxy pattern. There's white under the throat. And then on the right, we have much more white. So these are 2 different wolverines. And the one on the right, if you look in the box, she actually has teats. So we knew 2 things. One, that she's a female, and 2, that she is breeding. So we knew that she must have a den nearby. And this is Pepper the wolverine. I'm going to show a video in a sec here. And this is just outside to the east of Mount Rainier National Park. And we were able to locate that wolverine in the photo before, and we nicknamed her Pepper. And here she is at her den with her kits. This was in 2018. And this video marked the return of a female to the Cascades for the first time in 100 years. So this is her den in May. You can see it's a big old hole in the snow. The neat thing about wolverines is that they reproduce in the heart of the winter. So Valentine's Day is the official birthday of wolverines. And they dig their den straight down through the Cascades snowpack to the ground, and then they have their kits on the ground. They don't dig into the dirt. And so when we returned to the site in September, this is what it looked like. It was pretty amazing. It's this complex site with big boulders, caves in it, lots of down logs that were great protection for the wolverine. So you can see it's like a pretty neat spot for a wolverine to protect its kits safely. The detection of Pepper and her kits was quickly followed up the next year in 2019 by this image taken near Paradise inside the park. And so this is Joni, and you can see it's not as easy to see, but she also has teats. And so Joni is the first wolverine to reproduce in Mount Rainier National Park in a century. And we were wondering if we would be able to find her den, but that is a tall order. We never did find her den, but we set cameras in the park and we were able to get footage like this, showing her 2 kits. I think this was taken in June. So we knew, because they're so small and not very mobile, that the den must be nearby. And so knowing where the females are and where the den sites are allows the park to protect those places that are most important to building the wolverine population, to where the females are and where they den. Joni's a pretty neat wolverine. She's reproduced every year since we first detected her. So she's reproduced for the last 4 years. And here she is last year with her triplets. And you can see there's 5 wolverines in this video. So starting with the father and then the kits and their mom. The father's name is Van. And so it's pretty neat to have 5 wolverines all in one video in the park. And Joni reproducing 4 years in a row, that's not very common for a wolverine, and it really indicates to us the health, the intactness of the ecosystem at Mount Rainier and how it provides critical and important habitat for wolverines. So now I want to shift gears and talk about the Cascade red fox. So the Cascade red fox is one of the rarest species in Washington's Cascades, and it largely occurs from Mount Rainier to Mount Adams National Park. It is one of 3 subspecies of mountain fox that have a unique evolutionary history. They came into North America during the second to last ice age, and they persisted in North America during the last ice age. So they evolved and adapted to 100,000 years of glacial climate. This is an old map, but it was made in the 1980s, and it shows all the documentation of red foxes in Washington from fur trappers and from wildlife biologists. And what it shows is that along the crest of the Cascades in red is where you have a population of foxes that live high in the mountains. And then elsewhere in the blue, in the lowlands, you have this introduced population. And this was a theory and a hypothesis that was developed by my mentor, Keith Aubrey, in the 1980s, but it proved to be true based on genetic analysis in the early 2000s. And so we have these very different genetic lineages of foxes, and the Cascade red fox is the native mountain fox, and the blue ones, the introduced populations, they're the descendants of foxes that were brought west for fur farming and hound hunting operations, and then they either escaped or were released from enclosure. So this was sort of the picture of the Cascade red fox when we began work. We knew that they were at Mount Rainier, but not much about their status. And otherwise, there was just these 4 detections outside of the park. So it was totally unclear where the fox lived, and definitely how it was doing was really not known. The Cascade red fox, it lives high up in the mountain. It loves the subalpine parklands, all the area along timberline, and in the upper mountain forests where it forages on small mammals. However, we didn't know really which parts of the forest it relied upon, what exactly it eats, if any of the species that it eats are impacted by climate change. And at Mount Rainier, which has turned out to be one of the last strongholds of this fox, it's found in just a select number of subalpine meadows. Throughout the park, it's largely an east side species, so we find it right along the crest, and on the east slopes. It's rarely seen on the west side of the park. As Sallie mentioned, I conducted well, she didn't mention this, but the result of this work. So I conducted a deep dive into the genetics of the Cascade red fox across the South Cascades. I looked to see what its genetic diversity was, how big the areas that they occupied were, and if individuals were connected. Say if a fox that was born on Mount Rainier, was it connected? Did it to individuals on Mount Adams? And I did indeed find that foxes that were born on Mount Adams, they dispersed to Mount Rainier and vice versa. So there was good population connectivity, but where we thought we had multiple foxes, it turned out to be single individuals, and that the genetic diversity of the population is extremely low. And so this prompted Washington State to list the species as endangered that last year, and that's provided some new attention and support to understand what is driving this decline. So today we are shifting to understanding what is driving the decline of the Cascade red fox. We're doing it in a few ways. We're collecting scat samples and DNA metabarcoding them, which means that we're analyzing everything that is in the scat of the wolverine to see all the different prey items that it eats and identify which are the most important. And are those species being threatened as well? Are those species in habitats that need protection? Is things like climate change causing meadow encroachment that is limiting the area for the prey species to be abundant in? We also have a second project where we are working in collaboration with Mount Rainier National Park and with the University of Washington to fit Cascade red foxes with GPS collars and look how coyotes and other threats may be causing this decline. Coyotes are typically low elevation species, and with climate change and reduced snowpacks and also more outside of the park, but with increased recreation, coyotes are able to move up when the snowpack is hardened, either by rain occurring in the winter more often than historically, or by things like snowmobiles and ski tracks providing routes for coyotes up into the mountains. So we have just a little update. We have 3 foxes fitted with collars right now in and outside of the park, and we're working to get that number up to around 10 to 16. So I just want to briefly talk about our work in North Cascades National Park. I know this is all about Rainier, but we're doing some exciting work on Canada lynx, and I wanted to share that. So we went up to the North Cascades looking for Cascade red foxes to see if we could find another population separate from the Mount Rainier to Mount Adams connected population. And we collected 1,000 scats, and we thought we were maybe going to get some foxes and the odd wolverine and add to our database. But we didn't get any foxes or wolverine, but we ended up getting 300 lynx samples, and this converted into 70 individual lynx. And now 70 is not a large number, but it was a very hopeful sign for the status of lynx, because lynx researchers thought there was quite a lot less than that. And so now this was our minimum estimate, and now we're working to collect more data and convert that into a robust population estimate so we can see how many lynx there actually are left in Washington. And I just wanted to highlight one of our projects that I talked about briefly a minute ago, but this is our DNA metabarcoding project. So we're looking at, and this is where community scientists can really get involved. We collect hundreds and hundreds of scat samples every summer, and we send them to the lab to look at how the carnivores that live in the mountain ecosystems, how they divide what they eat. So if coyotes are threatening foxes, it might be through competition for the same food resources. And so the scat samples can tell us, is there overlap in their diets, or are they separate and not competing with each other? And just to wrap up, we have this vibrant community science program. You can hike collecting scat samples. You can also work from home sorting photographs or helping us with data quality control. You can be 1.5 years old, or you can be 80 years old. There's opportunities if you love to be out in the mountains. We also visit classrooms. We were in 14 classes in rural Washington and Oregon this school year, talking about the wonderful creatures in Mount Rainier National Park and the work that we're doing to protect them. I'd love to take any questions. Thanks so much.
Thank you so much, Jocelyn. That was great. I know I learned a ton and I'm freshly intrigued, and I'm sure that our audience feels the same. Now, folks, is the time to get any questions that you have addressed, or please feel free to put them into the Q&A box at the bottom of your Zoom screen. We're going to start out with a fun one on naming. You mentioned several names of the individuals that you've observed in the wolverine population. Who picks those names? How do they come to be?
Great question. Great question. So the Mount Adams Wolverine was Wildy, and that was a call out and a fundraiser. So in the early days, you could donate and suggest a name, and we picked our favorite Wolverine one. We've had a few of our colleagues that have come out to help us, or just sort of like a transfer of knowledge about wolverine biology that have named them. But this year, this last year with the triplets, we're going to put it out to some classrooms that we're visiting just outside the park so that kids get to select the names.
That's fantastic. Thank you. All right, and a two-part question on foxes and species interaction in general. So you mentioned that the foxes observed in Mount Rainier are mostly on the east side. Do we know why? As a follow-up, with all of these species, particularly the wolverines and the foxes, how are you seeing their interactions together? Has that changed anything for either population? Can you dive into that?
Okay. So as many of you know, foxes, they do that cool thing where they jump up and they pounce on the snow and they break through the snow, and they'll find the mouse that's in the subnivean zone under the snow. They are cursorial predators, which means they ambush their prey. And to do that, they need open forests and park lands. So the west side has more dense forest. This is what we think is going on. We are a little bit surprised. There's meadows on the west side. There's meadows on the east side. But they do seem to prefer the more drier, open forests on the east, sorry. But they are right up along the crest, and they do spill over a little bit on the west. They just do not go down into the forest, it seems. Oh yeah, and the interactions. So I do think it's funny to think of a little Cascade red fox and then this wolverine showing up back on the landscape. So these are species that have evolved together, but do not, haven't occurred together in recent times. We just have some very scant anecdotal information of wolverines following foxes. So we do, and this is through snow tracking, and so we do wonder and believe that a wolverine could probably kill a fox and predate upon it if it was lucky. But they're both very opportunistic feeders. They have like a broad diet, and so I wouldn't think that there would be a concern that wolverines are going to cause a problem or like lead to a population decline for the Cascade red fox.
Great. Thank you. All right, a couple more. We have more than we have time, which is a great problem. Wolverines, focusing back on them exclusively, what is their general range distance? How large are their territories?
So we know this from the North Cascades Wolverine study, which happened from 2005 to 2015. And the largest home ranges were 1,000 square kilometers. So they are ginormous. And Joni and her offspring, they roam around the entire volcano. So they're on the west side, the east side, the north side, the south side. They are outside the park. And then her mate, Van, his range goes almost to Snoqualmie Pass and almost down to White Pass. So they have extremely large territories that they defend, and one male's territory will typically cover 2 to 3 females. So as we get more females showing up from the north, hopefully in the coming decade, then they will probably set up their territories adjacent, but definitely not overlapping Joni's.
That's an incredible territory
Big.
size. That's amazing. Thank you. All right. What are the greatest challenges you've faced in doing this work? And What would make it easier?
Funding, more funding always. I mean, it is awesome work. It is not physically easy. I think some of the biggest challenges are getting stuck on your, well, outside the park on your snowmobile. Just get the Mount Rainier is a wild, remote, rugged place, as you guys know. And in the winter, it is very hard to access. So getting into the stations is a major challenge, but just having the staff capacity to run all these projects, it's always a matter of support.
Right. Fair. Thank you. Sure, that's the tip of the iceberg. All right. Focusing back on our fox buddies. You mentioned that you're observing them with GPS collars. Can you talk more about any new learnings from observing them via that mechanism?
This is brand new, so we don't have a lot of data yet. It's just been neat to see. We trapped 2 foxes at sunrise, and if you spent time there, you might imagine that the sunrise has the most incredible subalpine meadow complex. I always think when I go up there that this is all they need. They're just hanging out in this kind of zone from Sunrise Point, maybe over to Frozen Lake. But they have really large home ranges, and they're going way north, almost to Highway 410. So they're using really remote parts of the park, and we think we might have one denning, and it's really out there. It's nowhere near Sunrise, and that's been a surprise for me. But the fox collars are very small. I used to study grizzly bears where you had a big collar, big battery. You could get lots of locations. The fox collars, they take a GPS location every 6 hours. And so there's only so much we can learn about really their fine scale movements. But as the collars keep collecting data over the year, we'll learn more.
Thank you. Look forward to that. All right. And to bring it home, a couple on volunteer opportunities and upcoming events with you and your team. Specifically for the volunteer hikes to help collect scat for the community science. Can you talk more about locations, the physical nature of those hikes? How does that work?
Yeah. So you can reach out to us on our website. We'll be setting up a series of priority trails for Mount Rainier National Park and for the rest of the Cascades where we're looking for scats. The cool thing about this project and about Cascade red foxes and about Mount Rainier National Park is that the foxes are found in some of the most accessible parts of the park, like Paradise and Sunrise. We also need samples from some of the furthest reaches of the park. So we have a training video that Mount Rainier and us put together, and we send out sample kits, and we'll have these 2 in-person trainings in the park in June, and we'll teach you everything you need to know. And it's really just a matter of being observant, hiking through Cascade red fox and wolverine habitat and collecting the scats that you see.
Excellent. Thank you. And one more on upcoming events. It was mentioned that you have a speaker event coming up on June 19th, if I recall correctly, the Paradise Inn, Mount Rainier National Park. Can you tell us more about that event and how folks can learn more and join you live?
Yeah, so I'm excited to be coming to speak at Paradise Inn, so right at Paradise there. And in the next morning, we'll be going on a hike and looking for Cascade red fox and wolverine scats. The talk will be at 7 p.m. on June 19th, and it will be somewhat similar. It'll be fairly similar to this. Maybe I'll have some new updates on our research. But yeah, it'll be at Paradise, and the next day we'll hike out into the subalpine parkland.
Fantastic. Thank you so much. Before we wrap up, I want to say thank you again to Jocelyn. This has been amazing. Thank you to Sallie, also incredible Your support and participation to bring this project to our community is so deeply appreciated. This has been a ton of fun, and I can't wait to learn more about the project as the summer unfolds and in the years to come. This is great work that you all are doing, and thank you for sharing it with the WNPF family. For those who want to learn more about the project and keep an eye on it, again, we're joined by the Mount Rainier National Park team and the Cascade Carnivore Project team today. And I know I'll be keeping an eye on it and keeping in touch. I'm sure many of you will be too. Before this event ends, we also want to mention that today, May 8th, is the last day of Give Big in Washington. It's a great opportunity for you to support causes in Washington that you care about. And WNPF is asking our supporters to help us raise money for projects similar to the one you learned about today and more. Thanks to a generous donor who is matching gifts up to $10,000, your impact will go twice as far in Washington's national parks. You can take advantage of the match by visiting WNPF.org/givebig. Thank you to everyone out there for joining us for another virtual field trip. These are always wonderful events to host and share events in our community with you. If you'd like to see other episodes of our virtual field trips, you can always find us on our YouTube by searching Washington's National Park Fund for past virtual field trips and those coming in the future, as well as this one very soon. Lastly, if you're interested in joining us for future events, whether it's online or in person, visit WNPF.org/events. Again, as always, thank you to everyone for supporting our parks, the exciting work going on in these amazing spaces. And we look forward to seeing you in the future for another incredible virtual field trip. All the best until then. Thank you all.